to the PGC Worldwide Lab meeting. I see that people are still calling in, so let's give them a minute. Okay, looks like it's stable now. So today's topic is analysis across ancestry. We have 3 speakers today. We have Laramie Duncan, Elizabeth Atkinson, and Alicia Martin. All 3 talks are centered around GWAS analysis across ancestries, which is a really timely topic. Today we're going to do something a little bit different than usual. We are actually going to wait to take questions until all 3 talks are over, and then we will allow you to unmute yourself. So if you have a question for any of the presenters, then you can just unmute yourself and ask a question directly. And if that's not going to work, then we can still take questions via the chat function. Okay, so let's get started with Laramie Duncan, who is currently Assistant Professor of Psychiatry at Stanford University. Laramie received a joint PhD in neuroscience and clinical psychology from the University of Colorado, and then did a postdoc in statistical genetics in the labs of Jordan Smaller and Mark Daly at MGH and Harvard Med School. Today, she will be talking about cross-ancestry analysis in PTSD and the cross-population special interest group of the PGC. So take it away, Laramie.
Okay, great. Thanks so much, Caroline. I really appreciate this opportunity to present. So thank you to you and Pat. Like you mentioned, I'm going to talk about 3 topics today related to cross-ancestry analyses. First, the PGC PTSD analyses. Second, polygenic risk scoring analyses. And third, the cross-population special interest group within the PGC. So starting with PGC PTSD, the original wave of the PGC PTSD analyses was led by Dr. Kirsten Conan, and my postdoctoral mentor, as Caroline mentioned, was Dr. Mark Daly. They provided incredible leadership and mentorship for this, so I wanted to mention them up front. And the reason that we're talking about PGC PTSD is that actually compared to most large-scale GWAS, our samples were relatively diverse. So as you can see here, we actually had a minority of European ancestry participants, and we had a rather large sample of African Americans, as well as some Latino Hispanic individuals. This is, again, for the first wave of the PTSD analyses within the PGC. At the time when I made this figure, I looked at the other large psychiatric GWAS, and like the rest of medical genetics, we can see that most samples are of European ancestry. The samples that aren't of European ancestry across medical genetics tend to be from East Asian populations. So despite the fact that PGC PTSD does have greater representation of more populations, it's important to note that it's still not reflective of world ancestry. But nevertheless, in conducting these analyses, the fact that we had these more diverse samples necessitated modifications to our analysis. And so when we approached this problem, we did what anyone does. We thought about what might be appropriate, talked to experts, and we tried a number of different analysis approaches. And there were a lot of modifications that were necessary. Just showing one here, we decided ultimately to conduct a trans-ancestry meta-analysis. And so in order to do that, we needed to assign ancestry for each of the participants in our study, such that within each cohort, we could individually conduct ancestry-specific GWASs prior to meta-analysis within each ancestry, and then trans-ancestry meta-analysis. So actually, though, this work has continued in terms of making further analytical improvements to the pipeline. And so I just want to point everyone here to Caroline's work on this and Adam Mayhofer and her group. They've continued to improve the pipeline with some really nice additions and changes to quality control, imputation steps, and the analysis. And this second round of the PGC PTSD analysis is now available on bioRxiv. The link is here. So for questions about the most up-to-date improvements, I would suggest that people check out this paper or talk to Caroline. Going back to PTSD, the first wave of the PTSD analyses, though, I'd like to point out something that was interesting and informative from these analyses. So purely by chance alone, it happened to be the case that we had about 10,000 individuals of African-American ancestry and European ancestry, with about 25% cases each. Despite this these comparable sample sizes, it turned out that we had no significant results in the African-American samples, whereas as expected, we had significant SNP heritability estimates in the European ancestry individuals, as well as polygenic predictions. For example, using schizophrenia, external schizophrenia GWAS results to predict PTSD in the European ancestry samples. And based on power calculations, we thought that we would have enough power in these European ancestry samples, but due to expectations of lower transferability in the African-American samples, we didn't know if we would have enough power. And in these samples, it turned out that we did not. The reasons for this are at least somewhat well understood. Number one, there was worse coverage of African ancestry variants in our samples. This is both because African ancestry individuals have greater genetic diversity than other populations, and so for any given number of variants, you can't cover as much of the genome. But also, as many people know, there's a bias towards European ancestry variants on genotyping chips. So both of these problems probably contributed. Also, the lack of external data resources from non-European populations is really a problem. It's particular for these African ancestry samples, but this is true for other populations as well. And then finally, there are also methodological inadequacies currently. For example, differences in linkage disequilibrium and allele frequencies are still not handled as well as they could be in many different analyses. So the good news is that in the second wave of the PTSD analyses, this paper that I mentioned before from Caroline, we had larger sample sizes for all populations. And with these larger sample sizes, we could actually estimate heritability and have polygenic predictions in the African-American samples. So it does look like this was an issue of power. And so certainly, as expected, it helps to have larger sample sizes. I'm going to turn now to a couple of quick points about polygenic risk scores. So I don't know if it's partly just being in Silicon Valley and being near 23andMe, but everyone is talking about polygenic risk scores here. Not within my psychiatry building, but just people are very interested in these. And a couple of years ago when, for example, 23andMe was releasing results without any, or polygenic results without any mention that these scores might or certainly would have differential performance across ancestry groups, we thought that this was a really important topic to spend some time exploring. So to give a bit of background, as early as 2009, the ISC paper, Purcell et al., demonstrated that polygenic risk scores that are derived from European ancestry populations have poorer performance in African-American samples. And this poorer performance in non-European ancestry samples has been shown many times since in lots of different polygenic risk scoring publications. So what, kind of the new question that we wanted to look at was we wanted to quantify the decrement in performance between European ancestry performance and other ancestries. And we looked at a couple of ways of doing this. I'll just show one bit of information here. We looked at 10 years worth of polygenic scoring studies. First, to just see who was included in these studies. As expected, these studies also have primarily European ancestry, but then getting to the performance of polygenic scores across different ancestry groups. We made comparisons, and for example, one of the results is that we found that the polygenic scores performed about 3 times better in European ancestry individuals than in African-American samples. I don't know if, I think Alicia may or may not mention additional data that she has here that has, I think, even better analyses addressing this question. So in summary, regarding polygenic scores, the performance is worse in non-European ancestry populations. This is not surprising. This was, known for quite some time. But efforts are underway to quantify how much worse the performance is in different populations. I think it's important to know what to expect, both for scientific research, where we want to have accurate power calculations, and then to the extent that anyone is using these scores in clinical practice ultimately, or just, sort of in their own lives, getting 23andMe reports, it's important to know how well they might work. Efforts are also underway to improve prediction across populations. But getting to a bigger picture question, I think the big question that a lot of us have on our minds is, what do we do about the substantial underrepresentation of most populations in genetic studies? So this gets me to my last topic. And at first, or as a point of background information, there are many potential solutions to this problem, and I know that many researchers within the PGC have been working on this in this area for considerable time, and so we're just mentioning one additional approach. But we, if I had more time, I would mention other efforts that have been ongoing. So something that we did is that with Hai-Ling Wang, who is now junior faculty in Mark Daly's group at the Broad, we started a cross-population special interest group within the PGC with the goals of improving applicability of genetic results, conducting analyses, and supporting the involvement of researchers from different parts of the world. In one of our very first meetings, Roseanne Peterson had a really great idea to write a best practices paper describing the ways in which genetic analyses ought to be modified for different samples of diverse ancestry, and in particular admixed samples and just mixtures of different samples. And so some of the topics that are addressed in this paper include specific recommendations for how to modify quality control steps, which parameters should be modified and how and why. As well, there are recommendations for imputation and for how to analyze samples in a GWAS or mixed model context. And this is absolutely a huge team effort. I want to just say thank you to this group. I think that the reason that such a best practices paper doesn't already exist is that there actually really isn't any one person in the world that could have written a best practices paper. And it's really thanks to the analysts in this group, especially that have expertise in particular areas, that we were able to cover these different components of analysis and really write about the different approaches and what the pros and cons are for taking one approach versus another. So that's what's in this best practices paper. Hai-Ling spoke to an editor at Cell, and they were very interested in this paper, and so they suggested that we write it up as a primer. So that's the current format of the paper. We'll be submitting it on Monday, and so that's our first work product. And the special interest group is open to anyone. It's technically within the stats group, but if you're interested, we have meetings on the first Wednesday of the month at 1 p.m. currently, and you can get in touch with us. And just thanks to the group, all of these groups, for these incredible collaborative efforts. So thank you.
Thank you very much, Laramie, and thanks for being on time. This was a really nice presentation, and I'm sure that there are many comments and questions, but let's keep them until the end. We are now moving on to our next speaker. It's Elizabeth Atkinson, who is currently a postdoc at MGH and at the Broad with Mark Daly and Ben Neal. Elizabeth did her PhD at Washington University in St. Louis and is currently developing resources that allow for improved genetic analysis in admixed populations. Like Laramie, she's a member of the PGC PTSD group, and she has developed local ancestry analysis for us. Today, Elizabeth will be talking about her framework enabling well-calibrated genomic analysis of psychiatric traits across admixed populations. When the slides are up, let's see. Okay, I'll see them. So take it away, Elizabeth.
All right. Thank you so much again for inviting me to talk about this project. I'm really excited to hear the feedback. So yeah, as Caroline said, today I'm going to tell you about a project I've been working on, hoping to improve the ability to do statistical genomic analyses on admixed populations. So to start off, here we go. I'll reiterate a point that we're hoping is now becoming common knowledge in the GWAS community, and that Laramie just spoke deeply about. The vast majority of our association studies are conducted on European cohorts. And if we look more closely at this sort of wedge of the pie that is non-European, we'll notice that only a few handful of % are populations that are admixed. And just to make sure that we're all on the same page starting off, when I say admixed, I'm talking about an individual whose ancestry is not homogeneous, but rather is comprised of several ancestral populations. So importantly, there's actually many more samples out there who have been genotyped or sequenced alongside phenotypes, but they're not actually making it into this figure. They're being excluded from analysis due to being too admixed. And there's also several significant large-scale efforts to collect psychiatric phenotypes alongside genomic data in diverse populations, some of them led right here at the Stanley Center. So there really is a pressing need for the development of novel methods that will allow the easy inclusion of admixed people into association studies. We really can't afford to kind of leave large swaths of our data on the table anymore, and let alone the other problematic aspects of leaving entire ethnic groups understudied. So admixed individuals are generally removed due to the challenges of accounting for their complex ancestry, so that there are kind of concerns about population structure, infiltrating analysis, and biasing results, which can lead to false positive associations. Studies that do include admixed individuals tend to generally only correct for PCs. So PCs, actually, I think I can make a laser pointer here. Let me do that. All right, so PCs generally only correct for kind of average ancestry. So if somebody is, say, 75% African, 25% European, or vice versa, and they actually, can't really consider a lot of the more fine scale population structure that can still be present in the data. And this is important, which, this sort of fine scale pattern might be different, for example, among case and control cohorts, and still leave the door open for false positives. So here I'm going to present a novel analytical framework to hopefully rectify this issue and allow for the easy incorporation of admixed people into association studies. We specifically do this by accounting for local ancestry, which takes into account this finer scale population structure. So to get a little bit more into the actual kind of method, we're calling this method Tractor for the moment. The core feature of the proposed framework relies on inferring population structure, as I said, informed by local ancestry. So the first step is this automated pipeline to call local ancestry tracts in your sample. So just in case, you haven't seen a painted karyogram figure before, here's an example of a Latino individual. The autosomes are along the x-axis, positioned along the chromosomes on the y, and then the 2 strands of each chromosome are painted according to the ancestral origin of that tract. So we use this information after we collect it to kind of improve long-range phasing and haplotype tract recovery, which I'll go into in a lot more detail in coming slides. And then the end goal is basically to be able to extract out the component ancestries of interest. So for example, if you have a large European cohort and you have this admixed population, you could use Tractor to scoop out the European bits of your admixed cohort to include alongside your European individuals. And same goes for the African and Native American components in this example. So in this way, you can kind of leverage the information in admixed people. You don't have to exclude them from analysis anymore. So since the first step in this pipeline involves calling local ancestry, I wanted to validate that it was performing well on kind of the target populations that we had in mind. So the use case I'll be talking about for the rest of this talk is the African American demographic context, and we modeled this after PGC, PTSD, African American cohorts. So to test local ancestry inference using one of the very worthy existing methods to call local ancestry, RFMix, I simulated a truth data set that resembles these realistic PGC PTSD populations using real haplotype data. So this will retain the LD patterns and other genomic features present in real data that are sometimes hard to simulate. And using this truth data set with known phase and local ancestry, I then ran tractor step 1, calling local ancestry, and quantified how often we got it correct. And hearteningly, about 98% of the time, no matter how you slice it, we were getting correct local ancestry calls. So this seems to perform very well. Next, I wanted to see how the pipeline would work in kind of a more real looking data set. So usually we don't have perfect phase, we have statistically phased data. So I took our truth data set and ran it through a standard phasing pipeline, ShapeIt2 using 1,000 genomes as a reference panel. And we noticed something that we kind of hadn't initially expected to see, which is that a lot of the originally long tracts were really broken up by these switch errors in phasing. So even though local ancestry inference is performing really well, if it's calling well if we have 0, 1, or 2 African tracts at a given position, these long-range haplotypes are being disrupted due to switch errors. So we ended up building in an additional step in Tractor to find and unkink, is the word we're using, as if, unkinking this garden hose, to recover these long-range haplotypes. So here's our karyogram again now after this unkinking step has been implemented. We noticed that there were still sort of stretches where, due to the local ancestry, we painted the chromosomes on these statistically phased data, so there's some overhang areas that were still not able to be recovered. So we decided to implement one more iteration of RF mix on this sort of ironed out data set to see if this could help recover these full haplotypes. And indeed, this did dramatically improve the situation. So just to convince you that not only did this, make our karyograms look prettier, but this actually made the data look more realistic, I modeled the tract distributions using a Poisson waiting time centered at 9, which was the number of generations ago that the pulsive admixture occurred. So given this is what we would expect the European tracts in our data set in this, the European tracts of these African-American individuals to look like, given all things being correct. And we can use this to put a p-value on how likely it is to get the distributions in the various treatments of the data. So in our truth data set, again, focus on these red tracts, which is the European segments. We do see pretty close to expectations distribution here, almost around 9. After statistical phasing, however, the tracts get extremely short. They're actually p times 10 to the negative 28 likely to observe a distribution of this many tiny tracts. So very, very unlikely. After unkinking, it's about twice as improved. And with one iteration of the pipeline, we have gotten things almost back up to where they should be. And just sort of to zoom out and put this all into perspective, another way, you can see how we've really dramatically improved things from this original purple phased situation back down to this yellow line, which is as close to the truth data set in black here as we've gotten it so far. So we are indeed making the situation look statistically significantly more realistic by recovering these long-range haplotypes, which can be important too if you're concerned with things like LD, for example, in your data set. I've also tested this in a bunch of other demographic scenarios, and it performs, similarly well. So with different admixture fractions, putting a different, demographic models in, so pulse times at different points in the past, and also between different ancestries with varying kind of divergence times. So in the last couple minutes here, I'd like to kind of take a step back and look at one of the main applications of this framework, which is really to show that it improves performance in a GWAS context. So not only should we better correct for population structure by using this local ancestry information, but we can actually identify new loci through an increase in power from local ancestry as well. So the statistical model that's built into Tractor basically tests each SNP for an association with the phenotype using the logistic regression model that I show here, where X is the number of copies of the risk allele from the first of your ancestries. X2 is the number of copies from your second ancestry, and then you can put in whatever other covariates, importantly, including global PCs, as for the rest of your parameters. You'll also notice that this is currently written as a two-way admix context, but it could be scaled up to an arbitrary number of ancestries. And you can further test if the risk allele is ancestry specific by evaluating the difference between beta 1 and beta 2 using a z-test. So we wanted to see if there were any kind of power gains using this model. And I want to acknowledge Adam here, who heroically ran a whole lot of these simulations with me. So to do this, we simulated, again, a realistic African-American demographic scenario. We drew a biallelic disease variant with the probability of each genotype copy drawn from the minor allele frequency. We then simulated a disease phenotype, assuming a 10% disease prevalence, and an individual's risk of developing the phenotype was modified basically by their percent admixture and the presence of the minor allele on the African genetic background. So this is basically assuming no effect in a European genetic context, but an effect in an African genetic context, which would be, equivalent to modifying effect sizes due to a tag SNP being present in African background and not in Europeans for a shared causative mutation. So we ran a whole lot of simulations varying all of these kind of parameters to sort of characterize the landscape of how Tractor would improve your power. This is the simplest context, just showing the allele frequency held constant at a 20% minor allele frequency. And in all of these plots, the dotted line or the dashed line is Tractor, and the solid line is sort of the traditional GWAS method, which does incorporate PCs. And you'll notice that at both the sample sizes shown in blue and black, we do see a significant power gain using local ancestry. And this can get very large, and this gain can get really big in different genomic contexts. So for example, when you introduce a minor allele frequency difference between the ancestries, now shown in this red line, this sort of gap between the methods grows very dramatic. And I kind of want to dwell on this for a moment just to show how much, how significant this power improvement is. So if you pick your favorite power, let's say 80%, you'd be able to detect variants that have an odds ratio of about 0.1 lower using our local ancestry incorporating method compared to your traditional GWAS. Or if you pick your favorite odds ratio, let's say 1.2, you would basically have very little power to find this variant in a normal context, but we'd be able to detect it at very high power from using our method. So we've also done a lot of sort of tests characterizing other aspects of the genomic landscape. Since I'm running a little low on time, I'll just sort of briefly mention the most significant one, which is the effect size difference across ancestries. So now we've introduced an effect in both backgrounds, not only in the African genetic context. And the sort of major take-home point is that if you have the effect, so on the right-hand side here, we have the effect now swapped only in the European background rather than the African background. And you'll notice that we would have effectively 0 power to detect it using a traditional model, but can detect it at reasonable power with Tractor. This is, because European ancestry only makes up about 20% of the sample. So the signal would really have gotten swamped out if we hadn't deconvolved the local ancestry tracts. And importantly, in a context where we would actually expect no improvement from local ancestry, the effect is the same, everything is the same in both ancestries, we don't see much of a power loss, a very minimal power loss from using Tractor. So there doesn't seem to be, a major hit from including this in your analyses. So, if you're uncertain of the effect of ancestry, it seems like it's not a massive problem to incorporate this. And importantly, we're talking about the perceived effect of this tag SNP, not the real causative mutation. And so, even if there's the shared causative mutation is the same across ancestries, what you actually would detect in your GWAS could differ depending on the minor allele frequency or LD patterns or environmental interactions or many other factors that could be different across these ancestries. All right, to sum up, today I've talked about this readily implemented pipeline that should allow you to be able to include admixed individuals in your GWAS studies in a well-calibrated manner. I am currently optimizing it with members of the HALE team from the Broad Institute, so it should be a very quick and work in all systems once it's finalized there. But it's already written up in Python, and I can, distribute it to whoever's interested. We have also built in these extra features to improve long-range phasing, and I've shown you today that we not only boost your sample size by allowing inclusion of admixed individuals, but we can actually further boost your power by leveraging this information from admixed populations to find novel variants. We're also hoping to take this in a few kind of directions. I'm starting testing on an empirical data set that has a very well-established ancestry-specific hit, so blood lipids in African American individuals, to make sure this really does work not only in simulation, but in real empirical data as well. We're hoping that since recently admixed individuals have disrupted LD blocks, we should be able to leverage this to more kind of fine scale pinpoint causal variants. So we're sort of diving into this now too, and developing a heterogeneity test to try to automatically suggest and eliminate candidate sites based on your admixed populations. And I'm also working with Alicia Martin, who's speaking immediately next, and some other collaborators here at the Broad and MGH to try to build a polygenic risk score framework that would actually produce reliable estimates for admixed populations, which remains another kind of notable gap in current methods. And I'll just close with saying, not only is this procedure useful in the GWAS context, but could really be applied to any situation where you need to control for population structure at a very fine scale manner. So for example, even things such as, evolutionary studies doing genome-wide scans of selection. So with that, I'll acknowledge my collaborators and advisors, and I guess I'll take questions at the very end, but thank you for your attention.
Thank you so much, Elizabeth. That was really interesting.
Thanks.
Our third speaker is Alicia Martin. Alicia is an instructor at MGH and affiliate researcher at the Broad and Harvard. She received her PhD in genetics from Stanford University, and her current research focus is on developing novel statistical methods to improve the generalizability of genetic risk prediction from Eurocentric genetic studies. So her talk today is entitled Clinical Use of Current Polygenic Risk Scores may exacerbate health disparities. Go ahead, Alicia.
Thanks, Caroline. Let me, try to move this out of the way. Oh, my mouse is disappearing. Well, hopefully this goes away. So I'm excited to talk to you guys today, and thanks for having me here. Can you guys all see this part of the Zoom screen on the screen?
We can see your slides.
You can see my slides. Okay, perfect. So I think an important point to start at is in thinking about human population history. So all of our genetic differences across populations are sort of shaped by how we originated and how we migrated and mixed as we moved out of Africa. So of course, humans originated in Africa. This has been shown through genetic evidence as well as archaeological and linguistic evidence. So many sources showing this. And then humans migrated out of Africa. And as they did so, they took a subset of genetic diversity with them as they populated Europe, Asia, Australia, and into the Americas. Another point that I think is worth talking about, is the fact that GWAS are becoming super increasingly powerful. And so that's very, very exciting because we're making so many more biomedical discoveries these days. Some drugs are even being developed out of this. And this has been growing at such an exponential rate that it's been really, impressive and somewhat challenging even to keep track of all of this progress. So that's been really, really awesome to watch. Unfortunately, however, as Laramie mentioned earlier, and Elizabeth did as well, genetics has this diversity problem. So now shading the same growth and progress in genetics by the populations that are represented in these genetic studies at the individual level, you can see, of course, that the vast majority of participants in genetic studies, about 80% these days, are of European descent. And this is far out of step with the global population, where about 16% of the world is of European ancestry. And perhaps even more troublingly, if we look at the fraction of individuals that have been in these GWAS studies, as an overall proportion, the progress in diversifying genomics has somewhat stalled or perhaps slid a little bit since about 2014. And so this is a really big issue if we're trying to generalize studies to everybody. And so one of the nuances I want to mention here is that it's not that the studies have been getting smaller. Studies have generally been either staying the same or have been growing in different populations. But one thing that we've seen is that studies have been growing much, much, much more rapidly in European descent populations than elsewhere. And so one question that I really focus on is in understanding how do ancestry study biases in genetics impact the generalizability of the knowledge that we can learn from these studies. And so I break that down in a few ways in different aspects of our work, but in general, I want to highlight a few key points that I think are worth keeping in mind throughout all of these questions. So one of these is that the fundamental biology is really shared across different populations. So one person from one population will probably have, if they're going to have a heart attack, it's probably for the same underlying reasons, for example, as an individual of another ancestry. And that's true in genetic space as well. So when we've looked across many different biomedical domains and tried to understand what causal variant effects are in different populations, these really tend to be mostly shared. So it's not that there's anything special about genetics that's different from other biology. In general, the causal genetic variants seem to be the same and shared across populations. But there are some complications in interpreting genetics across populations for several different reasons. So of course, it's also worth keeping in mind that there's more genetic variation within than between populations. So populations are not substantially genetically differentiated to the point where we're finding completely distinct genetic populations. In fact, most of the genetic variation is shared. That's common across populations, and there's more genetic variation within than between. Another point that I think is really worth keeping in mind is that LD structure, this correlation structure of the genome, is one driving factor that's created a lot of challenges because this really differs across population as a function of human history, going back to the first map of that I showed you. So to try to address this question of how generalizable our genetic studies are, I started with computing polygenic risk scores. So Laramie talked about this earlier, this great enthusiasm in polygenic risk score space, and how it's been really impressive to see the growth in this area in the past few years. Just so we're all on the same page, I think most of you are familiar with a polygenic score, but in general, this is just predicting an individual's phenotype from genotype. So we're basically taking genotypes from some target individual, some effect size estimates from a GWAS that exists, multiplying these together, summing them up across the genome, and that's basically our phenotypic prediction. That's a very simplistic method. There are other methods, but some considerations that sort of cut across all of these different methods for computing polygenic scores are which SNPs we should include, what weights we should use, and one thing that we always need to address in terms of the utility of our polygenic scores is how accurate is the score. And this is really going to vary a lot with sample size, the heritability, the genetic architecture of the trait, and a number of different factors. So definitely worth keeping in mind all of these complexities in interpreting polygenic scores across the literature. So a study that we had done previously showed that population history really impacts genetic risk prediction across diverse populations. And so a few points here are that genetic prediction accuracy decays with increasing genetic divergence between the discovery and target populations. So in this figure on the right, I'm showing you a polygenic score distribution computed in the 1,000 Genomes project. And so you can see that we predicted European populations, for example, to be taller than American and South Asian populations, and we predicted East Asian and African populations to be the shortest globally. But these differences really don't line up with anthropometric studies, and they're rather misleading. These distributional shifts are really, really massive. And so this is clearly not necessarily reflecting reality. We also set up some coalescent simulations alongside some statistical genetic simulations and showed that these polygenic scores can differ across populations arbitrarily, and these are not necessarily meaningful. And we also showed that neutral human evolution alone can be sufficient to explain these differences. We don't necessarily rule out selection, but we do say that neutral evolution and drift in particular may be driving some of these differences. There's a really interesting couple of papers that I want to point out by Michal Sohail, Jeremy Berg, and colleagues that were published in eLife yesterday on this topic. So we wanted to look at this in a large scale setting. And so to do that, we looked at the UK Biobank data. The UK Biobank is, of course, mostly consisting of European descent individuals. And so we used those individuals to conduct GWAS for several different traits, 17 traits that were all quantitative, so things like height and BMI, and then a number of blood panel traits. And then we used the diversity in the UK Biobank in this non-European subset to try to understand how generalizable prediction accuracy is. And so in general, what we saw is that if we normalize prediction accuracy across all of these 17 traits to how accurately we predicted in Europeans, we saw a pretty substantial drop-off in prediction accuracy across these different populations. So for example, on the rightmost part of this graph, we saw a 4.5-fold improvement in prediction accuracy in Europeans with respect to how well we're doing in African descent populations. We're also doing about twice as well predicting in European populations as in East Asians. And you can see how well we're doing in these other populations. So these are really, really quite large disparities here. So why is this happening? A lot of people have written about this before, and it's been well covered in the literature, but in general, there's a pretty predictable basis of polygenic risk score disparities, and it obviously relates to who we're studying. So why though? Well, as you know, GWAS are best powered to discover variants that are common in the population. So if we're studying European populations over and over again, in general, we're detecting variants that are most common in European populations, which are then able to explain more of the phenotypic variation in European populations than those variants that are less common in other populations. Additionally, the LD differences across populations mean that we are probably getting better tag SNPs in the GWAS in European populations than in other populations. And then there are other really much more complex topics that are influencing this generalizability. So differences in environment, selection, and other more complicated differences. But I want to emphasize that there's a lot of consistent promise from diversifying efforts so far. So for example, the PGC schizophrenia working group, led by Hailiang Huang and with a first author, Max Lam, have been working to really increase the sample size of East Asian individuals in schizophrenia studies. So right now, the European case control studies in schizophrenia are still about 3-fold larger than the East Asian studies, but the progress in the East Asians have been really, really rapid and massive lately. And so when we looked at how well we're able to predict East Asian schizophrenia risk in these case control cohorts, in general, what we saw was that we were much better able to predict East Asian schizophrenia risk using the Ancestry matched East Asian data, despite the fact that the European training data was about 3-fold larger. So that indicates that there's really a lot of promise and value in doing Ancestry specific studies here. So lastly, I want to turn to this effort that we did to try to compare some biobank scale analyses. So generally what we're really interested in was doing equal size GWAS in the UK Biobank and in the Biobank Japan, where we have a lot of overlapping traits that have been deeply studied. And then we're interested in predicting in the European populations and in the Japanese population to try to understand what prediction accuracy looked like to see if this was symmetric and comparable across populations. We also tested prediction accuracy in the UK Biobank African descent populations. So in general, what we saw was that Ancestry matched disease prediction was most accurate. So on the left, I'm showing you along the x-axis the disease, and on the y-axis, I'm showing you the prediction accuracy. And the Ancestry matched results are indicated by matching colors. So on the y-axis is the target of prediction, and the summary statistics that we've generated, that we've used to generate the predictors are shown in blue for the Biobank Japan individuals and in red for the UK Biobank individuals. And so in general, we did better in both scenarios with the Ancestry matched disease prediction. one thing that's sort of interesting that we learned was that overall disease prediction was more accurate in Japan, and that was sort of a consequence of how these cohorts were constructed. So the Biobank Japan cohort is a more hospital-based disease ascertained cohort, whereas the UK Biobank is a healthier and wealthier than average population-based cohort. A similar finding emerges from the quantitative traits. So for these general health measures, anthropometric and blood panel traits, in general, the Ancestry matched quantitative trait prediction is also most accurate. Taking a look at these y-axes, you can again see notable differences in how accurate prediction is in each of these populations. And the quantitative traits are predicted most accurately overall in general in the UK European samples. And that's again because of this ascertainment on healthy populations of individuals. Okay, so I want to stop here and wrap up and think about next steps. So in general, I think polygenic scores are really exciting and interesting and may have some power to improve clinical models. But right now I'm a little concerned, or kind of a lot concerned, that they're likely to increase health disparities due to these vast Eurocentric GWAS biases. So I see this as a call for a few pushes that we really need to make concerted efforts on. One is we need much more diverse GWAS studies, and another is that we need new statistical methods to address these major issues. And on this topic, since it can be a bit sensitive, I just want to urge everyone that's working on these cross-cultural, widespread topics to communicate their research responsibly and widely, and anticipate the implications of your research, thinking about the potential negative consequences that will happen. I also wanted to point out that there are large efforts, for example, going on in Africa to scale up GWAS studies there, especially in psychiatric space for example, developmental disorders and for schizophrenia. And I think this is really exciting, and it's taught us a lot about how to do cross-cultural research in an ethically responsible way that accompanies some capacity building efforts. So I want to acknowledge that this is a huge effort involving a ton of really awesome people, including my advisor and mentor, Mark Daly. And with that, I'm happy to stop and have everyone take questions, I suppose.
Excellent presentation, Alicia. Very interesting. Thank you so much. All right. So, Tammy, can you make sure that that people can unmute themselves?
Yes, it looks like they can, and they can also ask questions in the chat box or the Q&A box at the bottom of their screens.
Okay, let's see. Are there any questions? So I see people raising their hand. Let's see. Segun, would you like to go ahead?
Okay, so thank you very much. My name is Segun Fatumo. So today I'm calling from University of South Florida under Monica. So I have many questions, but I will just ask the most important one to me today. So I have one question each for each presenter. But my first question goes to Elizabeth. So I want to ask Elizabeth the method, the design, which is called Tractor, and she has implemented this method by evaluating this method with the common way of checking for population structure. That's the principal component. I wanted to ask Elizabeth, have you also compare your method with other model that is used to control for population structures, such as a linear mixed model, which was implemented in many software? So I would like to know how if your model is much more better than the linear mixed model, which is commonly used now in Africa to do GWAS.
Sure, yeah, thanks so much for your question. So yeah, we started off kind of doing the simulations with a case control framework. And I guess it's also important to note that in all the comparisons that I showed you today for Tractor versus the traditional GWAS model, we do include principal components in both. So that is, improvements even beyond the sort of incorporation of global ancestry. This really is like a local ancestry specific gain. But yeah, we're also hoping to kind of think about other, phenotype contexts beyond the case control framework. So yeah, some things more to be done, but I think we should expect similar power gains across contexts. If that answers your question.
All right. Next, I see Leah Davis.
Hey, can you hear me?
Yes.
Yeah. Okay, great. Thanks so much. These were great presentations. And I guess, so one question, we've been struggling with this in the context of the biobank in terms of performance of polygenic risk scores, and trying to think about ways to potentially squeeze out more information from the European-centric GWAS that would be translatable to non-European populations. And one thing that we've thought about but haven't yet tried, and I'm curious if any of the panelists have tried this, is instead of taking a clumping approach that's based on p-values, which will be in part driven by the allele frequency in the discovery sample, using a clumping approach that's basically based on the effect size, so that you're retaining the SNP with the largest effect, but also the highest allele frequency in the target population. And I'm wondering if that could possibly, of course it's not going to solve the problem, certainly, but could it possibly improve the predictions a little bit? Anybody looked at that?
We've looked at it a little bit, Leah. So I guess in terms of two different things, both in terms of prediction accuracy, and then also in terms of, Alicia showed the slide where the different 1,000 genomes populations have apparently different distributions of polygenic risk scores.
Mm-hmm.
So when we clump on all ancestries instead of clumping on European ancestry, and when we use a smaller set of SNPs that accounts for a greater LD across all the populations, we actually see pretty much normalized distributions across all ancestries. So that was kind of interesting. We tried to look at how well the predictions worked across ancestry groups, doing clumping ideas like you mentioned. And I think, unfortunately, in what we looked at, we didn't have enough power to really determine if it was making a difference. So I can't say for sure, but it seems like a great idea to look into inadequately powered data sets.
Yeah, adding to what Laramie said, there are a bunch of different approaches with how you would do clumping and how you would potentially evaluate even in, sort of other methods. And they can, various methods can improve on prediction accuracy across populations. So for example, LD-pred is a little bit better than sort of a pruning and thresholding approach, but not as much as we would like. And so one issue I think is that there's no method currently that I'm aware of that looks at multiple LD panels across populations to try to translate sort of the effect size, the marginal effect sizes from one population to a marginal effect size to another population. And so that's an area that we're actively working on developing new methods in. And so stay tuned, and hopefully we'll have some new method that you can try out there.
Yeah, awesome. Well, if you are looking for a biobank size test case that with a lot of diversity, let us know.
That's great. Thanks.
Great. All right, we can take a few more questions. I see David Curtis.
Yeah, hi. So I guess people know me. So two things really. One is where I'm coming from is that I think it's quite likely that polygenic risk scores will never ever be clinically useful. So with that as the context, my question is really, don't we think there's a risk that in African populations, because of the structure, the diversity and the lack of linkage disequilibrium, that the kind of phenomena that we see with GWASs in populations that have been through the out of Africa bottleneck in East Asia, in Europe, that stuff just won't work in African populations, that polygenic risk scores will not work in African populations, or would require at least sample sizes of 10 times, 100 times what we're using to generate polygenic risk scores now. And I think the question is, if polygenic risk scores are going to be clinically useful, are we going to massively invest in getting these huge African samples and seeing whether they work or not? Or are we at this point going to look at it carefully and think maybe they won't be clinically useful and therefore we'll do other things in Africa, but not that?
Yeah, Dave, that's an interesting question. And we have also been kind of thinking along the same lines, like how would you improve things for everybody the fastest? And so clearly the way that you would improve things for everybody is by starting to look at the most genetically diverged group from what's already been studied the most. So looking at the most genetically diverged group from Europeans means looking in Africa. And so the way that we would generally help African populations have higher prediction accuracy, East Asians, South Asians, admixed populations, everybody would be to start in Africa to build up these very, very large cohorts. And so I think that's part of the reason we've had such a concerted effort with trying to push much larger studies in Africa, because it's been so vastly underrepresented in genetic studies. To kind of your first point, will they ever be useful? So I mentioned super briefly that there is some promise from diversifying efforts. So the Ancestry matched results do a whole lot better in non-European populations than just using a larger European population, even when they're several-fold the sample size. And sort of a meta-analysis approach might improve things even more. So I think in general, having a very, very large African cohort is probably like step one, and then diversifying and meta-analyzing a lot of different groups together is probably step two. And yeah, you're right, we're a long ways off, but I think that's kind of the direction we need to move in.
Thank you, Alicia. That kind of also feeds into the question that Gita Senthil posted. What kind of sample sizes do we need across various populations to compute ancestry-specific PRS? So do you have any idea of, it's probably very difficult to say, right?
Yeah, that'll depend a lot on genetic architecture of the traits you're looking at, how shared the genetic, what the genetic correlation across ancestries is. Yeah, it's a hard question to answer without some more specifics, unfortunately.
For sure, some more funding would help, right? All right. So I see we have time for one more question. Monica, do you want to?
Sure. Excuse me. Can everyone hear me?
Yeah.
Great. Well, first of all, I'd like to compliment our three presenters today for doing a fabulous job, and in particular, bringing to a larger audience our attention on this very important problem that, it sounds like you've individually and collectively been working on for some time, but sometimes I don't think the larger scientific community is as plugged into this as they need to be. So you're doing a great service today by presenting what you've been working on so well. My question is, I think, really for Elizabeth, and in particular in relation to something that Laramie mentioned. So Laramie mentioned that our abilities to sort of detect these ancestry differences was informed a little bit by the PGC PTSD's more diverse cohorts that included a larger proportion of individuals from African ancestry in particular. And so these methods have been developed using those data, if I understand correctly, that Elizabeth presented. Will those methods be available soon for other PGC cohorts to use, or even for other cohorts that aren't focused specifically on psychiatric outcomes?
Sure. So thanks so much for the compliment to us all and for the question. Yeah, certainly. So we're using, PTSD in an African-American demographic context as kind of our initial use case, just to test the performance of this method, but it certainly could be applied to any complex phenotype. It's built around psychiatric phenotypes, so certainly psychiatric phenotypes, but even kind of more broadly in medical genetics, complex traits in general should be all served well by Tractor. It should also perform well, provided local ancestry performs well, it should perform well in other populations. So I'm starting to do some work looking at more complex demographic scenarios. So for example, three-way admixed Hispanic Latino individuals are a little bit of a trickier situation than a two-way admixed population. So I'm scaling it up now and making sure that it'll still perform well in those contexts as well. But yeah, basically any time that you can accurately call local ancestry, which depends mostly on divergence between the component populations, Tractor should be able to handle it.
And so do you have a timeline for making the code available? I know you said we could email you, but to deploy it on the Broad website or something like that.
Yeah, I'm working with some developers for this, Hail software, which is hosted by the Broad Institute and is, going to make it a lot fancier and run faster and work in every computational environment. So I think the timeline for that is certainly within 6 months. It's currently a set of Python scripts that runs, but probably will run nicer once I get the Hail developers to pretty it up. So within 6 months, there should be a bioarchive paper with the software attached to it, I would say.
So,
Excellent. Thanks so much, everybody. I think we are past the hour now, and I would like to thank all the speakers and all the many participants. This was a really interesting World Lab meeting. Thanks so much.
Thanks, Caroline.
Bye.
Thanks, everyone.
Yeah, thanks so much.